Alright, so we're going to go over the questions in chapter 15, and I'm going to try to emphasize what I think is most important about these questions. The first one is, what is the one gene, one enzyme hypothesis? This were experiments carried out by Beadle and Tatum back in the 40s, looking at mutants in Neurospora. And they had these mutants. I'm going to need a little more room than this. So they had a particular type of mutant, and so they would grow these things on plates. They actually use little tubes, but you can do it on plates too. And so that's a petri dish with some media in it. Here's another petri dish with some other media in it. And this is complete medium, meaning that it has all of the nutrients that the the fungus needs. And this is minimal media that is missing a bunch of stuff. And so they found these are colonies growing on this plate. They found some mutants that's a colony, and then we'll put a colony here. And so the mutant that was here, or this guy, can grow on the complete medium, but it can't grow on the minimal medium. And so that means that, so on minimal medium, so this is sort of like, if you think about even in humans, there are certain amino acids that we can make ourselves, and then there are some that we have to eat in our diet. And so this plate is providing all of the amino acids, and then this is missing amino acids. And so a wild type normal cell can make some of its own amino acids. And so in this case, there's a mutant that can't make something. And so we know that it can't make something because of these wild type guys. So these are wild type. And then this guy that used to be here is over here. He's a mutant. And we know he's a mutant, and the phenotype of the mutant is that he can't make something. So that type of mutant is called auxotrophic. And this is just, I just want to make sure you get these terms down. Auxotrophic mutant means it can't make something. We don't know what it is that it can't make yet. And so this is a mutant, and then the wild type that can make everything that it wants to make, or that it needs, is called prototrophic. So we got auxotrophic and prototrophic, and the auxotrophics are the mutants, and the prototrophic is the same as wild type. So what is it that this thing can't make? Well, we can figure it out by adding stuff back to the medium and see if we can rescue that mutant. So now if we take that mutant that can't grow on minimal medium and we add stuff to it, let's say we take that minimal medium, and what they used is the amino acid arginine. None of these pens really seem to work very well. Minimal plus arginine. This is as good as I can get this thing to write. Then it will grow. So now the mutant will grow. So it won't grow on just minimal, but if you add arginine, then it will grow. So that means that that mutant is somehow defective in making arginine, because if you add arginine, it can grow. And so if we have a biochemical pathway of a bunch of things where we're going to make compound A and convert it into compound B and convert it into compound C and then eventually convert it into arginine, if we have a mutant in one of these steps, we've got a mutation so that we can't make arginine, that mutant can't grow so let's say we just have a mutant here. We'll just call it mutant 1. And we know that it can't grow on minimal, but it can grow on minimal plus arginine. So there it grows. Now this particular mutant, we could also, so let's say mutant 1 is there, and let's say we have another one. Another mutant that can't do this step. And so mutant 1, so both of them, if we add arginine, then they'll be able to grow. Mutant 1, all we have to do is add whatever this is, this intermediate on the way to making arginine, and it will grow. Because if we add B, we'll have bypassed this step that is necessary, and it should be able to grow. Just wanted to make sure I was recording. All right, so this is just to give, this is kind of a long answer to this question, but I just wanted to give you this idea of how these things work. So we're going to have some different mutants. We'll have wild type. We'll have mutant 1 and mutant 2. And then we're going to, up here, we're going to have stuff that so we have just minimal media. We'll have minimal plus A. These are different things that we're adding to the medium. Minimal plus B, and then minimal plus arginine. Now wild type yeast, or in this case Neurospora, these are all on different plates. So wild type can grow on everything because wild type can make its own arginine. So we'll put a plus for growth. So wild type can grow on everything. These two guys are mutants, and we identified them by the fact that they can't grow on minimal. And they can also grow, we also showed that if we add arginine, they can both grow. And that tells us that the mutation is somewhere in the pathway that makes arginine. But now if we look back up at our where the mutants are blocked, so mutant 1 is blocked at the step going from A to B. So as long as we add anything that's after that step, then that thing will be able to grow, because that mutant is deficient in the ability to make B. So if we add B, it should be able to grow. But if we add A, it won't be able to grow because it can't convert A to B. So mutant 1, if we add A, it still can't grow. But if we add B, it can grow. So that tells us that mutant 1 is at the step between A and B. Mutant 2 can't grow if you add A because it can't convert it to B. And it can't grow if you add B because it can't convert B to C. But you would be able to, mutant 2 would be able to grow if you added compound C. So that's just a brief, so mutant 2, if we added compound C, it would be able to grow. So mutant 2, if you put it on something that had C in it, so minimal plus compound C, then mutant 2 can grow. Because C is after the step that's defective in mutant 2. So that's a colony of Neurospora fungus growing. So I'm not going to ask you to interpret these kinds of tables on this exam. This takes a decent amount of practice to get the hang of it. But I just wanted to give you a sense of the type of experiment that was done. And what the key to this step is that by figuring out which step each mutant was defective in, so mutant 1 is defective at this point, mutant 2 is defective at this point, each of these steps is catalyzed by an enzyme. So an enzyme, there's an enzyme that will catalyze the conversion of A to B, and there's another enzyme that will convert B to C, and this mutant is specifically defective in enzyme 1, and this mutant is specifically defective in enzyme 2, and a mutant means that you've mutated a gene. Now they didn't even know what genes were. All they knew is that genes were something that you could mutate and get a phenotype. And their big sort of advancement of the genetics field was that this particular mutant is mutated for this particular enzyme. So that's the one gene, one enzyme hypothesis. One gene corresponds to, come on, help me out here. One gene corresponds to one enzyme. So that was the first step in correlating genes with proteins. They knew genes were proteins. I mean, they knew enzymes were proteins. But they didn't, at this point, they didn't even know if genes were DNA or not. They just knew that a gene was something that you could mutate and get a phenotype. And now this, adding the one more piece to the puzzle, was that each gene coded for a particular enzyme. Now we know that each gene codes for a particular protein, and even later on, we know that one gene codes for a particular RNA, which may or may not code for a protein. But this was a historical experiment that showed that each gene coded for a particular enzyme, and so it was the first thing to correlate genes with proteins. And they didn't know anything about DNA yet. Alright, so now this one, let's get rid of some of this stuff. So that's why I don't go over that in this online format, because it takes too much practice, and you have to put in a lot of time to get the full measure of that. But if you practice with it, you can get it. This one's pretty simple. What is the significance of the fact that many synonymous codons differ only in the third position? So what that means is, so if we look at our genetic code, which I have one here somewhere. So if you look at the genetic code, you'll notice that, let's say, phenylalanine, UUU, UUC, UUA, UUG. Well, for phenylalanine, UUU and UUC code for phenylalanine. For leucine, well, leucine's not a good example because there's some other differences. Let's look at serine's a pretty good example. So if we look at serine, we got 4 different codons that can code for serine, and they all differ at the third position. So that's one significance of it, is that you can have different sequences in your RNA, and they'll all code for the same amino acid. So if you're looking at the messenger RNA, and you have a bunch of codons, and let me go back and see what serine was. Serine is UC something. So if we have a codon that is UC and then something else, no matter what that other thing is, we'll get serine will be put in there. And then the anticodon for this will be A, G, and whatever is here in the anticodon. So this anticodon is part of the tRNA, and it lines up to the to this position of the mRNA, and no matter what is at this third position, it's going to put in a serine. So with that, also another significance of that is if you have UCA, UCU, UCG, or UCC, all of those give you serine. So if your normal sequence is UCA and then you mutate it to UCU, you'll still get serine. So oftentimes a mutation in the third base position of a codon will result in the same amino acid, and that's called a silent mutation. So that's usually a mutation in the third codon, but it's not always the case. So if you see a mutation that's in the third codon, don't immediately assume it, because look over here, if we have UUU, that's phenylalanine, and this UUA is actually leucine. So in some cases it's not true. So whenever you get a mutation like that, you have to double check the genetic code to see if it's a silent mutation or not. So chapter 15 is mostly about the genetic code and translation. And for some reason, when I write on these things, this kind of stuff happens. Alright, so this is just asking things about the genetic code. Again, I don't like to go too much into the historical stuff here about how Nuremberg and all these people figured out the genetic code. And that's interesting stuff, but it happened in like 1945 and we got to move on. So a lot of people like to go through all the history. Alright, so reading frame. So reading frame, I think you all have a pretty good sense of what the reading frame is. You have a mRNA, and that mRNA gets read by the ribosome, and it gets reads in groups of 3, and each group of 3 is called a codon. And if you read all the codons that code for a particular protein, then that's the open reading frame for that particular gene. All right, so we all know what an open reading frame is, and that sort of thing. But it's not quite that simple, because in any given DNA sequence, there's a lot of potential reading frames. So if we just have any kind of sequence here, A, C, G, T, A, so etc., etc., we'll make this a double-stranded DNA. And this would have a 5' end and a 3' end. And up here a 3' end and a 5' end. Now if we don't know anything else about this sequence, let's say we just took this chunk right out of the middle of a gene and we were wondering what the open reading frame was. There's several possible open reading frames. So if we started the first one, we could read AAC, then GTA, then TGC. So that's one possible reading frame. We could start at the second nucleotide and go ACG, TAT, GC, and then it would go on to whatever the next sequence is. That's another possible reading frame. We could start at the third nucleotide and go CGT, ATG, and then C, and then down from there. That's a third possible reading frame. So we call these reading frames. This would be the +1 frame. This green one would be +2. And then this black one would be +3. Plus meaning we're using the top strand, and we just call that the positive direction. So those are 3 possible reading frames. However, we don't know, a gene could be transcribed either left to right or right to left depending on where the promoter is, and then that's going to make an RNA, and that RNA can be translated. So we could easily, without knowing anything else, we could easily transcribe this DNA sequence from right to left. So that means that if we did that, if we went right to left, then this bottom strand would be the RNA-like strand, and we would have 3 possible reading frames on that side as well. So if we started at the first nucleotide, we call it -1. Start at the second one, call it -2. And we can start at the third one, so this A down here, and start reading in that direction and call it -3. So in any given random DNA sequence, there are 6 possible reading frames, 3 going forward and 3 going backwards. Now, which one is the real one kind of depends, and you have to try to figure it out. In this particular example, it looks to me like maybe here's an ATG, and so that might start the open reading frame going left to right. Now it kind of depends. If we looked at a bigger sequence, there would be several possible ATGs we could choose from, and we'd have to decide which one is right. But if we just look at this, we look at that ATG, and that ATG happens to be in if we look here, there's ACC, so that's not it. So this is our reading frame, and which one of these reading frames was it? It lines up with the black one, so it's the +3 reading frame. So if we start this reading CGT, then ATG, that's where the actual translation would start. Alright, so that's the idea behind what reading frames are. Once we get a gene and we know where the reading frame is, so whenever we draw genes out like this, and we say that this is the open reading frame, so there's an RNA with a 5' sequence and a 3' sequence. And if we looked at the DNA, this would be ATG in the DNA. And maybe it stops with a TAA or something. Alright, so that would be the open reading frame. It's open because you got several codons. There would have to be a bunch of codons in between those. And if it's a long open reading frame, that's likely the open reading frame for that gene. But there's 6 possible reading frames, and the open reading frame is really the true reading frame. So the initiation codon is just the ATG that starts translation. No sense going too crazy. In RNA it would be AUG. Termination codon, that's where translation stops, and so that's termination codon is like UAA, UGA, and UAG. Now, you don't really have to memorize those because you'll always get a genetic code, and it'll show you in the genetic code that that's what's going on. And so over here we've got, where are these stop codons? So UAG stop, UAA stop, and UAA, UAG, the other one is UGA, so that's over here. Stop. It doesn't matter this ochre, amber, opal business, don't worry about that. That's just some old-fashioned lingo. So a sense codon is any codon that codes for an amino acid. And that's it. So all those codons in the table that code for an amino acid, those are called sense. And a nonsense codon is one that doesn't code for an amino acid. So a nonsense codon is the same as a stop. And so when the ribosome sees a nonsense codon, it says, oh, wait a minute, that doesn't make any sense. I don't have any tRNAs that will fit in there and give me an amino acid, so that doesn't make any sense, and so that's nonsense. And so when we talk about mutations, there will be nonsense mutations where we convert a we convert a sense codon into a nonsense codon. All right, and how is the reading frame of a nucleotide sequence set? And that's just by the AUG. So if we've got 6 possible reading frames, we've got a double-stranded DNA, and we don't know where the reading frame is. Wherever, and so there's going to be 3 possible ones. Sorry, it's a prime. So that's kind of like back up here, where we looked at these 6 different things. How do we know which one is the right one? The ATG or the AUG that's in the RNA will set the reading frame, and then from that point on, the ribosome will read every 3 letters, and each 3 letters is a codon. And so a lot of times you'll be given a DNA sequence and say, here, translate this, and there's going to be 6 possible open reading frames that you could translate. Which one do you use? You use the one where the start codon is. So the start codon will set up, and if there's multiple start codons, then there's like a whole process of figuring that out, and that's actually where this idea of gene annotation comes in. When you get an unknown, or you get a sequence and you don't know where the gene is, you have to figure out which ATG to use. We call that annotation, because you're actually going in and investigating it and figuring out what the best open reading frame to use is. All right. How are tRNAs linked to their corresponding amino acids? This is just so there's a bunch of enzymes in the cell that will recognize a particular tRNA. So here's a tRNA. Whoa, it's really hard to draw tRNAs with this pen. All right, so we'll just pretend that's tRNA. Yeah, let's try that again. All right, so we have a tRNA. Looks sort of like that. So that's a tRNA. Transfer RNA. But it doesn't have an amino acid on it yet. And so there's enzymes, and there's a whole bunch of these different enzymes that add amino acids onto there. And they add the proper one on, so whatever tRNA this is, it gets a particular amino acid and gets added there. And it actually, so this is like 5'. Somebody might ask you this, probably not me. So it gets added on to the 3' end of the tRNA. So this is one RNA, it goes around, it has this secondary structure, and then you get an amino acid added there. This is a tRNA. The one on the left is tRNA. This one has an amino acid on it. So it's an amino acyl. So that just stands for amino acid, amino acyl tRNA. That's just a tRNA with an amino acid on it. The enzyme that synthesizes an amino acyl tRNA, so the enzyme that does this reaction, is an amino. I'm going to type it because I can't write to save my life here. So the enzyme that does this is an amino acyl tRNA, tRNA synthetase, because it's an enzyme that synthesizes amino acyl tRNAs. You can have specific ones. So if you want to make a glycine tRNA, you would have a glycyl tRNA synthetase. If you're going to make phenylalanine, you would have phenylalanine alanyl tRNA synthetase. Another couple of terms that are used to talk about these things is that this is an uncharged tRNA, and then once it gets its amino acid added to it, it's a charged tRNA. And so the enzyme that does that is there are all these specific tRNA synthetases that recognize a specific tRNA because they have a slightly different structure and a different sequence, and they can recognize that and add an amino acid to it. How does the process of initiation differ in bacterial cells and eukaryotic cells? Mostly eukaryotic cells use the 5' cap, meaning the ribosome recognizes the 5' cap. In bacteria, the ribosome recognizes the Shine-Dalgarno sequence. And then one other thing that's a little bit different in bacteria, the initiating amino acid is a modified form of methionine called formal methionine, or FMET. So you'll see FMET as the initiating amino acid in bacterial translation. I don't think it really makes that big of a difference, but that's what it is. Let's see. And so what does that look like? So just in eukaryotes, you have a 5' cap, and the ribosome binds that and then cruises along until it finds the first AUG. That's in eukaryotes. And then in prokaryotes, you have this internal sequence called the Shine-Dalgarno sequence, and that's where the ribosome binds. And then it finds the AUG after that. So I think those are the major differences. Let's see if there's anything else that I'm missing. Yeah, that's about it. The other thing that the book points out at this point is that there is a sequence here associated with the start codon in eukaryotes called the Kozak sequence. That's the word sequence. In the Kozak sequence, that's just a consensus sequence. There's additional nucleotides that surround the start codon that make that start codon a good start codon to initiate translation. There might be other start codons so AUG is going to show up all over the place, and only some of them are good start codons. And the good ones, if you compare all the good start codons that you know function as start codons, and you look at them, there's a particular sequence around them that is somewhat recognizable. So if you're trying to figure out which one, if you have an unknown gene, and you're trying to figure out which start codon is used, you can look for that Kozak sequence, and that would suggest that the one that has the better Kozak sequence is the one that is used. What brings about the termination of translation? You have to hit a stop codon. So you've got your ribosome cruising along, and it gets to a stop codon. And you've got your tRNA in here and your protein coming off of it, and then the ribosome comes up to the stop codon. This is going to be the A site, and we'll come back to these sites in a minute. And the A site usually the A site gets positioned over the next codon, and an amino acid gets added. But this is a nonsense codon because there is no tRNA that recognizes this particular sequence. So you have this other thing called a release factor, that comes into the ribosome at that particular point and causes the cell to stop translating that particular RNA. And then there's several different release factors and GTP involved. I'm not worried about all those details. Just that there's a release factor that binds into that ribosome, and then the whole thing falls apart. So the RNA gets released, the protein gets released, and it'll eventually fold up. The ribosome lets go, and these ribosomes, the small subunit and the large subunit, they can go now and they can go find another RNA and start translating again. After translation, proteins get folded up. So what are some of the things that happen to proteins after translation? So there's a lot of things, but the basic idea is that after translation, we've got a protein that gets released. And so now here's our protein. The protein has to fold up. So post-translational modifications are things that happen after translation. Post meaning after. And so one thing has to happen, it has to fold up. But then you can have different modifications. You can have things like phosphate groups added. You can have sugar added. You can have this thing called ubiquitin gets added, which is another small peptide. Lots of different things can get added. I would say that the two most common that you might see are phosphorylation and the adding a sugar. So we've got phosphorylation, which is just adding a phosphate. That's very common. And the idea here is that you make a protein, but maybe you don't need it quite yet. You just want to make it and you want to have it around in case you need it later. And so the protein will be inactive until it gets phosphorylated. So you have your protein, it's inactive, then you add a phosphate, and now it's active. So it's very common. Adding a sugar is called glycosylation. So the prefix glyco refers to sugar. So glycolysis is the breakdown of sugar, and glycosylation is the addition of a sugar. There's lots of different sugars that get added, and the biochemistry of glycosylation is pretty complicated. And if you remember anything about carbohydrate sort of terminology, it gets complicated super fast. But sugars often get added to the surface of, not to the surface, but to a particular amino acid in a protein, and then that can somehow modify the function. Glycosylation is common to proteins that end up on the cell surface, and if you look at any picture of a cell membrane with all the proteins out there, there'll be a lot of glycoproteins, which are sugars that have sugars that are added to proteins and that are sticking out the surface of the cell. Oh, let's see what else they got. Let's see if they all right. So I think those are the two examples that they give. The main thing that you know is that there are these different types of post-translational modifications. Know one or two examples, and that should be good for now. Crap, I wasn't recording. Alright, so numbers 16 and 17 were originally on this list, and they're kind of detailed problems about those sort of Beadle and Tatum type things for the one gene, one enzyme hypothesis. We're not going to worry about that too much. Certainly go over that, and when it comes time to maybe studying for the MCAT or something, that historical stuff might show up. But for the most part, if you just get the general idea that the one gene, one enzyme hypothesis was looking at those types of mutants, those auxotrophic mutants that are mutations in particular steps in biochemical pathways, and by adding back the substances that are made by those pathways, you can figure out exactly which step in the pathway is affected by a mutation. And that's how Beadle and Tatum came up with the idea that genes somehow related to enzymes. They didn't really know much about the details. Referring to the genetic code, give the amino acids specified by the following bacterial sequence. Ooh, bacterial. So let's see if I'm going to grab this whole thing, and you have to have a genetic code when you're looking at this thing. So let's look at our genetic code, and we'll put it here. Alright, so if we look at this, we've got this one starts with an AUG, so let's assume that I'm going to rewrite this so I can manipulate it a little bit. So AUG, UUU, this is an easy one, AAA, UUU, AAA, UUUUGA. By easy, I mean that because it's UUU and AAA, I actually know what some of those are off the top of my head. So you guys should have a decent amount of practice already translating this, but the more practice the better. So if you look at this, and if we say you're going to start at the first AUG, there's an AUG, and then you can kind of offset it every 3 at that point and separate out what your codons are. And so I happen to know, so here we're going to put in a methionine, which is our first. But if it's bacteria, then it's going to be F-methionine. And then UUU is phenylalanine. Then AAA is lysine. Then UUU is phenylalanine. Then AAA is lysine. Whoops. And phenylalanine. Now the reason they're using these particular amino acids is that when they did when they figured out the genetic code, they made these RNAs that had just a bunch of U's and a bunch of A's, and that's kind of how they figured out the genetic code. But again, that's a historical thing that I don't think is all that critical. Just be able to use this genetic code. I think a lot of these questions refer to the genetic code. All right, so this was just FMet, and then it was going to be phenylalanine, lysine, phenylalanine, lysine, phenylalanine. So that would be the answer to that one. Now I like to use the single letter code usually if I can, but always put in the M at the beginning. A lot of students will put just the word start, but there's no proteins that have the word start at the beginning. They have a methionine at the beginning. And then don't put in any stop codon. Just the N, it just ends with the last amino acid. I mean, the stop codon's there, but don't add the word stop or anything at the end of the protein. So a non-template strand is shown what amino acid sequence will be made by this sequence. So this is a non-template strand. So let's make it a little bit bigger so we can deal with it. And if that's a non-template strand, we're going to need the template strand. Oh, but we don't need the template strand. So non-template strand means, so non-template strand is the one that we want, but the template strand would be this. but we don't really need that. All we need is this template strand, or not this non-template strand. So this is the template. So template for transcription is what they're talking about. And so then if we were to transcribe this, the RNA is going to look just like that. But where there's a T, there will be a U. So this is a lot easier to do if you're using a computer than just doing it yourself. So you can just copy and paste the non-template strand, and that gives you your RNA. Now most of the time we don't even bother with that. If we're given this strand right here, we can just translate that directly and just in our brain convert these T's to U's. So let's grab that sequence. That's the sequence we want. Look at our genetic code table. Get rid of this other stuff. Say that's where it's going to start. So let's separate out our codons. And this little stretch is going to be methionine. Then ATA, now we got to use our brain to convert this. So ATA is the same as AUA, and that's an I. Oops. So I for isoleucine. Then get down here to CTA. Up here that would be CUA. CUA is L. AGG is A is the first letter. G is the second letter. G is the third letter. That's arginine, and that's R, so it's not A. Then CCC, I happen to know that one, is proline. Miller. So it doesn't really mean anything. And so in this case, they didn't give you a stop codon. They just said just translate this little bit. Easy enough. Then they give you a bunch of other types of things that just to kind of get you used to and give you some practice with using the genetic code. And so we can go through some of those, and it kind of gets to the idea that there's multiple sequences that can code for the same amino acid and that kind of thing. So we have MET, TRP, HIS. Now there's only one codon for MET, so that has to be AUG. TRP. Now TRP actually is another one that, let's make this a little bit bigger so we can see what we're doing. So MET, TRP, HIS. Just got to remember that. So we want to figure out the code for MET, TRP, HIS. MET is always AUG. TRP, it turns out, if we look on here and you look for tryptophan, where is it? It's usually over here. Okay, here it is. There's only one possible codon for that. It is UGG. Alright, then HIS. Histidine, is there only one for histidine? Oh, so this is the easy one. So histidine, there's only one. Nope, there's two. So there's CAU and CAC. So there's CAU, so that's one possibility. And then the other possibility is AUG, UGG, CAC. So there's two possible sequences that could code for that little thing. Now you can see that gets pretty complicated pretty quickly. If one of those amino acids was leucine, there's 6 possible codons for leucine, and we'd have to write, and if you had 6 possible codons for leucine and then 4 possible things for proline, you have to come up with all those different combinations to tell you all the possible sequences. So if you're given the amino acid sequence of a protein, it's not super straightforward to work your way back to the nucleotide sequence. There's usually multiple thousands sometimes of sequences that can code for that particular amino acid stretch. Okay. And then this is just more amino acid practice. Which of the following amino acids could result from a mutation that changes a single base? And this is just to practice the playing with the genetic code. All right, so so the question is, of all of these things, which of these can there could be just a single base that would make this change. So let's just do them one at a time. Try to do it relatively quickly. So if we want to go leucine to glutamine, so leucine was one of those that had a bunch. Glutamine, where's glutamine? So glutamine's over here. So glutamine is CAA to CAG. Leucine, so glutamine is, so that's sort of the easy one because there's only two, so CAA or CAG. Now leucine is all these other possibilities. So leucine is CUU, CUC, CUA. So if we take leucine, one of the possibilities for leucine is CUA. If we just change that middle nucleotide from a U to an A, we get we go from leucine to glutamine. So yes, we could do a one nucleotide change to go there. So go from phenylalanine to serine. Alright, so phenylalanine, there's only two. There's UUU and UUC. Serine, there's UCU, etc., etc. So UCU is one. So we could go from UUU to UCU. That's one change. Goes from phenylalanine to serine. Answer would be yes. Phenylalanine to isoleucine. So now we're going to go from phenylalanine to isoleucine. Isoleucine has, what do we got? Three possible ones for isoleucine. So we've got AUU. Alright, there's our answer. If we just change that UUU to AUU, then we changed one thing and we went from phenylalanine to isoleucine. So yep, we can do that. Proline to alanine. Proline, I know CCC is proline, so CC anything is proline. So let's say CC anything is proline. Alanine is in here somewhere. Arginine, alanine is GC anything. So as long as we change that first letter from a C to a G, then we can change a proline to an alanine. Asparagine to lysine. Might as well do these last two. So asparagine to lysine. You guys get the idea. Asparagine, and it's good to just kind of go through this because that way we can say all these amino acids. Asparagine, aspartic acid, arginine, it's got to be around here somewhere. Asparagine, oh, there's only two. So AAU or AAC, and lysine is just AAA or AAG. So AAA or AAG. This is one of those cases where if we change the third letter of the codon, we actually get a change. A lot of times that's silent, but here we can just change the third and change asparagine to lysine. So yes, and then they asked you to say whether it was the first, second, or third, but we did that, but I just didn't write it down. And then isoleucine. I think this was isoleucine to asparagine, so ILE to asparagine. And isoleucine could have 3. So we've got isoleucine is AUU, AUC, AUA. And asparagine, I think we only had 2 of those, AAU and AAC. So AAU or AAC. And if we took this AUU and changed that middle one from a U to an A, we would change it. If we took AUC and changed that middle U to an A, we would change it. If we took AUA, we can't really change that with just one change. But yes, so all of those things, just one letter change can change one amino acid to another. Now, while I've got this genetic code, we'll come back to this later, but you'll notice all these color codings where nonpolar, polar, basic, and acidic, those are your 4 major types of amino acids. And so if you have a nonpolar one, which is yellow, so let's look at this example. We've got isoleucine, which is yellow. That's a nonpolar amino acid. Asparagine's over here in this sort of light greenish, and that's polar. So we're changing a nonpolar to a polar amino acid. I just threw that genetic code table in there so we'd have one. And then we're just sort of thinking about how translation works. And here's a picture of a ribosome in the process of translation. And you need to label a few things. So in this case, the RNA is always read. Now this is sometimes a source of confusion. I go through some efforts to say that the sequence is always read in the 3' to 5' direction, and the new sequence is made in the 3' to 5' direction. But when it comes to translation, we're not converting one nucleic acid sequence to a new nucleic acid sequence. That rule has to do with the 3' hydroxyl group on the nucleotides. In this case, now we've got the ribosome actually moves across the RNA in the 5' to 3' direction, and it starts to link these amino acids together. You can see this is bacteria because it has an F-met to begin with, and it asks you to label these things. So 5' end is to the left, 3' end to the right. The A, P, and E sites, so whenever you draw a ribosome, you have a small subunit. I always draw it with just 2 sites because they're really the main ones. And so these are the P site and the A site, and that's where most of the action happens during translation. It turns out there's another site over here where at the very end of, well, actually as each tRNA is leaving, it hangs out in the E site for a moment. So these days, at some point in the last 10 years, they added a 3rd site. So we've got E, P, and A. And so this would be E. So it would go E to the left here, P in the middle, and then A to the right. The start codon is the AUG. The stop codon is way down here, the UAG. Amino and carboxyl ends of the newly synthesized polypeptide chain. So that's this guy. So we're making this protein right here. And during the process of translation, the way that translation works is, if we've got a ribosome, and we've got a tRNA in here, and we've got an amino acid on there, and then we've got another tRNA in here, and this thing is growing. So as translation occurs, we keep adding amino acids on. And what actually happens during translation in this next step is this whole chain this thing gets taken and added to that thing. And the direction that this goes, and we'll look at amino acid structure in a minute, but there's a carboxyl end of the protein, and actually I just did that wrong, so let's undo that. There's a carboxyl end and an amino end, and when you're doing translation, the amino end is here. So you have NH2 there, and you basically make the protein from the N terminus to the C terminus. So when you eventually make this whole protein, you're going to have all of these amino acids linked together, and the first amino acid is the N terminus, meaning it has the amino group at this end, and down here it has a carboxyl group. And so we call that the N terminus and the C terminus. And so if we were to label this, this is the N terminus, and then down here is the C terminus of the protein, and that's at the very end. All right, approximate location of the next peptide bond that will be formed. It's going to be formed right that, well, it's actually going to be, we're going to have another tRNA, charged tRNA, so it's charged, meaning it has an amino acid on it. So that next one's going to have to come in here. It's going to have an amino acid on it, and then this is going to form there. So I guess the approximate position where that's going to occur, the peptide bond will be formed right in this area here. And placed on the ribosome where release factor 1 will bind on the ribosome. I don't know, probably here. So eventually we're going to get, the ribosome is going to get to here. And we'll have a tRNA in here. And then we'll have this protein that's coming off with all these different amino acids. And then the next thing to come in here, we'll make it red for stop, is a release factor. And apparently it's release factor 1. But just any old release factor will be fine. And so definitely go over the process of translation and watch videos and see how that works. What will be the anticodon of the next tRNA added to the A site of the ribosome? The anticodon is going to be, so the next codon is ACG. And so, we'll type that one. So the next codon is ACG, and that's 5' to 3', because that's the way we're reading the codons. Make this a little bit bigger so you can see what the heck's going on. And so when the tRNA matches up, it has to match up in the 3' to 5' direction, and then it'll have a U, A, C, and that'll be going in the 5' direction. Now this is part of the overall tRNA, but this part here is the anticodon, and it's matching up with the codon. This is variable, so ACG, whatever that code's for, that is variable. We could look at the genetic code, so ACG is a threonine, so that last letter could be any one of these 4 nucleotides. So we could just say X. So the peptide codon could have any, let's say N, that's what people use. So N means any nucleotide. What will be the next amino acid added to the growing peptide chain? Well, this is our codon, ACG, and I just said that that was ACG is threonine. So the next thing to be added would be threonine. Just sort of taking that genetic code and putting it into context with the actual ribosome and what's going on at the ribosome. This is just another example of that, and here I decided to throw the genetic code in so we could do it. So I wonder if I can copy this sequence. Probably not. So let's see what we got here. Let's take this. And move it down here. So I don't know if this is eukaryotes or whatever, but this is from a figure in the book where showing this in the context of the ribosome. So we have methionine, CCC, I happen to know, is proline, ACG. So what I would do if I was given this sequence is I would go through and say, okay, well, if this is my start codon, whoops, well, somehow bracket off the 3. And you can just kind of somehow write on there that what's what, and then maybe just write these things up here. So methionine, proline, then ACG. ACG is threonine. ACU is also threonine. GCG. GCG is alanine. AGC is serine. GUU is valine. CCG is proline. CCG-CUA is leucine. AGG is arginine. And then it stops. And so you don't put anything here. You could, like, as you're going along translating, maybe put a little dot there or something. But the actual amino acid sequence is just this. In what ways are spliceosomes and ribosomes similar? In what ways are they different? This one is pretty simple as far as I'm concerned. So the main thing I want you to know is that they both have, so spliceosomes have small nuclear RNAs, so snRNAs plus proteins. Ribosomes, so these are spliceosomes, and then ribosomes have ribosomal RNA plus proteins. And so they're both these sort of large complexes with a bunch of proteins. And they also have RNA in there. So somehow there's RNA in here, and maybe there's an RNA over here. And ribosomes also have RNAs in there. So the main thing is that they both are comprised of, or made of, RNA and protein. So that makes them both ribonucleoproteins. In fact, that's what ribosome means. Ribosome means that some means body. Any big structure in the cell is called a something some. You got proteasome, ribosome, chromosome, big things that you can see under the microscope. And so the ribosome is a big thing that has ribose in it. RNA is part of it. So they're both ribonucleoproteins. We'll quick check the solutions in the book. And this is number 40. I lost my mouse. There it is. All right. So number 40. Yeah, they go into some more detail, but it's not really that big of a deal. So know that they both have RNAs and proteins. The ribosomes in the cytoplasm, the spliceosomes in the nucleus, that's a big difference. And what they do is different. The spliceosomes involved in splicing and the ribosomes involved in translation. So I've got a little bit more to add to this chapter in terms of this stuff. General amino acid structure. I guess I could do this really quickly. This is turning into a pretty long thing. So I'll actually come back to that. So I'm just going to go ahead and post this video so that you have the main thing, but also go in and do this. Do this on your own. Draw a general amino acid structure. Maybe we can do it real quick. So amino acid structure has an amino group, has a central carbon, and it has a carboxylic acid group, and then it has an R group here. So that's a general amino acid structure. That R group is going to vary, and that's what makes the different amino acids different. A peptide bond is you take two of these things, so you've got an amino group. So a dipeptide is two amino acids put together. And the best way to draw it is just to draw them both out. Then this would be a different amino acid with a different R group. And it would still have this carboxyl group. So this is a carboxylic acid group. We're going to lose a molecule of water and stick these two things together. So now this C is connected to this N over there. We lost some water. We've got a double bonded O. And then this H actually comes down here. We have this, that's a C with the R group and the C double bond OH. And then over here we're going to have a C with the R group and the NH2. So there's a dipeptide. We've got our N terminus. And down here we've got our C terminus. And if we link a bunch of these together, we make protein. And so this is a peptide bond. All right. The four major types of amino acids, we can get that from our genetic code. So you don't have to memorize this because you can use whatever info you want for tests, right? So, but you need to know the significance of this. These different amino acids have different chemical features. The yellow ones are all nonpolar. The blue ones are polar. The kind of greenish ones are, well, I guess they're kind of blue-greenish or polar. Which ones are polar? Let's see here. Oh, so the kind of light green, bluish are polar, but they're polar and uncharged. The purplish ones are basic, means they're positively charged, and then the red ones are acidic, meaning they're negatively charged. So let's go over that over here. So we're going to have polar, but uncharged. So that's things like serine. And so we'll have an R group. So we have our amino acid, and then we have an R group. And serine has this structure. And this is polar, because that's like an OH group, hydroxyl group in water. That's polar. You could also have, oops, I was trying a new little feature there that didn't work out so good. You could have charged. So polar uncharged is different than charged. So charged, you could have the first obvious one are those that have a carboxyl group, and those are acidic groups. And then they can get ionized to form this kind of a group, and this is negatively charged. And so this is an acid which becomes negatively charged in the cell under physiological pH. And the positively charged ones are considered bases, because they can actually take up an extra proton. Oops. And these are positively charged. So both of these things can be polar uncharged or charged, but they tend to be charged under physiological pH. So these are positively charged, these are negatively charged. And then your nonpolar ones are just nonpolar. So if you have a side group, like a methyl group, for example, and if you look at all the structures of these different molecules, you can see that the nonpolar ones, maybe it has, all they have are carbons and hydrogens, maybe something like that. And so those are nonpolar, they're non-charged, and so they're nonpolar, which makes them hydrophobic. All right, and then the other part of this was understanding the different the different levels of protein structure. And I'm going to go over it relatively quickly because you all know this from previous class. Why when I do an empty page does it keep putting that on there? Alright, so our different levels of protein structure. Primary, that's just the sequence of amino acids. Secondary, where we have some local folding. So this is secondary. And there'll be some hydrogen bonds in between here. So in between here, so if these are our amino acids, the bond in between those are peptide bonds. Secondary structure, we're going to have in between these folds, these local folds. These are supposed to be dotted lines I'm doing here. These are hydrogen bonds. So secondary structure is primarily hydrogen bonds and van der Waals forces, which are just sort of weak interactions between things. And then a couple of other, and then it's going to fold up into a tertiary structure, as this. And if you have multiple polypeptide subunits, we call it the quaternary structure. And these are held together by all kinds of bonds. So we've got H bonds between polar amino acids. So you might have two polar guys like this. And you'll have a hydrogen bond between there. Ionic bonds between a negatively charged and a positively charged amino acid. So like aspartic acid and lysine. Aspartic acid is an acid, it's negatively charged, and lysine is positively charged, that's an ionic bond. Non-polar interactions. They don't really call them bonds, they call them interactions. Or hydrophobic interactions sometimes. And so if you have a protein and you have water on the outside of the protein, all your hydrophobic guys are going to be in here, like isoleucine, leucine, phenylalanine. A lot of your hydrophobic amino acids are going to be inside the protein away from water. And then the last type of bond that you'll see, there's one example. So peptide bonds are covalent bonds, but when we're talking about holding together the three-dimensional structure, so we're talking about bonds, if we've got our big three-dimensional structure, we're talking about these kinds of bonds between R groups in the protein. There's one example of a covalent bond, and that's called a disulfide bridge. Disulfide bridge, and that's between cysteines. So a cysteine side group, I think there's a carbon there, but then there's an SH. And you have another one that has an SH. These tend to form a bond like this. And this is called this disulfide bridge. So it's a bridge between two sulfurs, and it's called a disulfide bridge. So it's important when thinking about the genetic code that you know which types of amino acids are which. Now, you don't have to memorize it, but you need to understand that if you change a non-polar to a polar, that's going to mess it up. If you take a polar amino acid that's involved in a hydrogen bond and you change it to polar, it can no longer, or to change it to non-polar, it can no longer do hydrogen bonding. If you take a aspartic acid and you change it into a lysine, you're taking a negative amino acid and changing it into a positive amino acid, and that's going to be a big change. And so I'm going to leave it at that for now, but I just wanted you to review that.